Hello, ladies and gentlemen. Thank you for joining our Poultry World exclusive webinar in cooperation with our partner Innovad. We are a little late due to some technical issues, but they will be worked out as we go forward. My name is Fabian Brakter, and as the editor-in-chief of Poultry World, I am the host of today. In our webinar, we will focus on chronic gut inflammation in broilers. For the modern producer, chronic low-grade gut inflammation has a negative impact on the productivity of their operation. And often it stays under the radar, subclinically. But it's definitely felt economically as it impairs the bird's ability to absorb nutrients, grow and reach its genetic potential, which is huge nowadays. In this webinar, I have two speakers for you who will share their cutting-edge research on this important topic, a novel model to demonstrate chronic gut inflammation, and of course, how it can be tackled. The speakers are Michael Kogut and Jason Loger. Welcome to you both.
Thank you very much.
Good to have you on board. As you can see, our speakers are joining me via live video feed, as although the corona measures are eased as time moves on, it's still somewhat difficult to travel to the studio all the way in the Netherlands. And it's stormy outside as well, so planes, not so good today. We are broadcasting live, which means you can interact with us during and directly after the presentations. If you mouse over your screen, an orange Ask a Question button will appear, where you can type the questions to our speakers. And I invite you to use this function. Before we go to the first presentation, I have a poll question for you at home and in the office. The results will give us a nice talking point at the end of the webinar. So, your votes, please. The question is, what percentage of broilers do you believe are affected by chronic gut inflammation? And you can click on the answer of your choice, which is A, less than 10%, B, less than 25%, C, up to 50%, D, over 50%, or E, all broilers, equaling 100%. So please click on the answer of your choice, and we will come back with results at the end of this webinar. Well, let's kick off with the first presentation. Our first speaker is Michael Kogut. Michael is lead scientist and research microbiologist at the Southern Plains Agricultural Research Center in the US. And his research is centered on poultry gut health and alternatives to antibiotics to control disease and increase production. Today he will share his latest insights in the relationships between antibiotic-free poultry, low-grade chronic inflammation, and gut health. Michael, I'm looking forward to your presentation.
Thank you, Fabian, and thank you to Poultry World and Innovad for the invitation to speak this morning. If I can get the first slide, I appreciate it. What I'm going to do today is give an overview. The first half of the talk be an overview of this relationship, to talk about why chronic inflammation is becoming more and more of an issue, why it wasn't in the past. And in the second half of the talk, I'm going to give some new data that, full disclosure, was done as part of a PhD project by Gabriella Del Pont, who's nearing the end of her PhD right now. So if I can have the next slide, please. Basically, what I'm going to do is going to break the talk down into four components. I'm going to start off talking about my definition of gut health and what it means, and it laid the groundwork for the rest of my talk, as well as a sort of an introduction to the second presentation. Then I'm going to transition into this idea of AGPs. What has happened with AGPs? What did they do to prevent inflammation in the past, and why their removal has caused this increase? And then I'm going to again transition and talk about inflammation as a whole, and want everyone to at least walk away with from today's webinar with the idea that inflammation is not always negative. It's a connotation right now when someone says inflammation, it's a bad thing. We're going to talk about the bad things, but inflammation is a necessary physiological response, and we'll talk about that. And then the last part is I will show this new data, a relatively new data that Gabriella found, where one of the issues with studying chronic inflammation was developing a model and or defining some biomarkers for chronic inflammation. Next slide, please. So let's first of all talk about gut health. Let's define gut health, and so that everyone is on the same page. So let's break it down into its components. The gut. What is the gut? I think everyone understands that the gut is a tube. Most of us think of the gut as only the intestine, post-stomach down to the vent. Realize that the gut really starts at the back of the oral cavity. But for today's talks, we'll just refer to the gut as the intestine. It is the largest interface between the environment and the bird. So keep in mind, the lumen of the gut is an extension of the external environment. It is not the inside of the bird. The gut is a barrier. It separates this external environment from the inside of the animal. And because of this, the gut has the largest number of immune cells in the body. We always think of maybe the spleen, the bursa as being major cell sources. But the gut has the largest concentration because of the interaction with these outside environmental components that they may be exposed to. And lastly, the microbiota. The gut provides a habitat for the microbiota. And I think we all are aware of what the dynamic of what the microbes in the gut do and how they perform and their usefulness. Now let's look at the other side of the equation, define health. Now the dictionary's term for health is either absence or avoidance of disease, which is great, but I think when we're talking about a chicken in a chicken house, maybe disease is a little too harsh, but it's tough to get away from the fact that there's a lot of stresses, environmental stresses there. I like to define health as, if we go to the next, please. I like to define health as the ability of the animal to be able to perform its physiological role. That is, in other words, can it withstand these stressors, these environmental stresses that occur in the chicken house? And if they do, how close to 100% of genetic potential can the animal perform? The closer to 100%, the healthier the animal. And I think as we go through today and we talk about chronic inflammation, we realize that maybe that 2 or 3 or 4% that the birds aren't quite reaching 100%, that just an increase of a small percent, less than 1% increase in performance means millions of dollars worldwide to the poultry industry. Next slide, please. Now, when we talk about a healthy gut, there are various functional elements that we need to be aware of. And this is important because I'll refer to this later on. Now, this is under homeostatic conditions. Obviously, the gut is the place for food digestion and absorption. So we have to have that in place. Again, I bring up the microbiota. It provides that environment and allows the microbiota to perform. And a stable microbiota is important. Regardless of what the bugs specifically are, they need to be stable. The immune system obviously has to be functional. This barrier system, and this is a barrier system. It is microbiological, it is physiological, it is chemical type of barriers, and they must be in place for a healthy gut. I think a lot of us forget that there are cells within the gut and below the gut that are neuroendocrine, that produce hormones, growth hormones, and or the enteric nervous system that talks to the brain, and the brain talks back to the gut to help control the physiological aspect of the gut. And lastly, the oxidative status of the gut is important, especially for the function of not only the immune system, but of the microbiota. The stability of oxygen and oxygen levels in the gut are important, and I'll refer back to this later on. Next slide, please. Okay, let's transition now and talk about antibiotic growth promoters. There's been controversy, and still is, as to what did they do? What allowed AGPs to be considered growth promoters? How did they allow the animals to grow? And as I said, there's been some controversy, some other ways. Some people say it had an effect on the microbiota. Some people say it prevented pathogens from growing. Keep in mind, AGPs were used at sub-therapeutic levels. So the idea that they were protecting against pathogens is a little bit naive. I think the most amount of data that is out there is that AGP provided an anti-inflammatory effect, that they kept the stresses that are provided or the animals are exposed to were dampened by the presence of AGPs. And there's I cite 3 papers, there's more, where this shows that was shown that the AGPs had this anti-inflammatory effect by reducing mediators such as cytokines, but by producing materials that were anti-inflammatory. So they kept the local environment down. Now keep in mind, we're talking local at the gut level, not systemic. And there was an effect on the microbiota composition and the load, but keeping the good bugs, the bugs that were immunologically important, to a level, to a homeostatic level. Next slide, please. And once the AGPs were removed, this is the result of what we have now. Now, inflammation. Inflammation right now in the poultry world and the animal production world seems to be a 4-letter word. First, let's realize one thing. Inflammation is the effector mechanism of the innate immune response. I'll think back to your immunity courses. Immunity is made up of 2 components, the innate and the acquired. The innate is what you are born or hatched with. It does not need to have a antigen response. It will respond to whatever is presented to it. So it is a normal physiological response that is highly coordinated. And I think this coordination is important as we go on to keep that in mind. All information involves energy. It takes energy away to activate and keep immune cells functional. So there is going to be energy removed. Inflammation inevitably will cause a little bit of damage. And again, depending on how long it is, how much of that damage occurs. Its sole function is to get rid of whatever's causing it, repair the damage that's there, and return to homeostasis. Okay. Now, what is the cause? Well, I call it a trigger, and the length of the inflammatory response depends on the trigger, and that trigger can lead to multiple types of inflammation. The acute response obviously is very rapid. It's recognized, the trigger, whether it be a pathogen or some sort of tissue injury, is recognized, it's removed, resolved in a matter of days. So the energy that is expended towards it does not really take away from the animal's production because it's very, very short. But a chronic inflammatory response means that the trigger is constantly being exposed. And the biggest component of a chronic inflammation is the diet or a stress, heat or cold stress, for example. But the dietary components are made up of many, many immunological factors that will keep an inflammatory response going. And if you're using a diet that has a immunological component in it every day, no matter what you're doing, that trigger is there. So it's difficult to get rid of the inflammatory response if that trigger is constant. Next slide, please. Now, this slide, just for everyone's information, and it's interesting when we think back to the poll question at the beginning. It's not just diet. There are multiple environmental factors that can play a role, can act as triggers, depending on the management practices of a farm. And I have a list of some that are here that can actually be stressors, that can be triggers, that could lead to both acute and or chronic inflammation. And you can read through these and see that there's some acute, some are can be chronic type of things. But it's very, very important to realize that we can't pinpoint just one and exclude it, that these multiple factors can cumulatively induce an inflammation, and that we have to, if we're going to try to get rid of an inflammation, we have to keep in mind there are multiple factors. Next slide, please. Now, this slide I just put up here, and Jason's going to talk more about this as we go along, but Why is chronic inflammation a problem in the industry, and how does it affect performance? Well, to feed the immune cells takes away nutrients and energy from the animal. It takes a lot of energy for these immune cells to function, especially at the innate immune response level. These cells need a lot of energy. And so they're taking that energy away from the animal, and that animal then is going to lose performance because it's feeding the immune system and not feeding the animal. Next slide, please. Now, I ask you to remember, these six factors, the functional elements involved in homeostasis. Well, what happens in an inflamed gut? What changes are occurred looking at these functional elements? If we can have the first one, please. So in terms of food digestion absorption, there's a decrease in enzymatic activity. There's a decrease in nutrient transport to the animal. It's going to the immune cells. It's not being directed towards the animal. Obviously, the microbiota is going to change. You're going to change the stability. You're going to change the diversity. And just to give you an example, there's a decrease in small chain fatty acids, which I'm sure everyone realizes is very, very important because it is the energy source of the epithelial cells that line the gut. If you're decreasing the small chain fatty acids, the epithelial cells are going to be not as functional because they're not being provided the energy that they have. Obviously, with the immune system, you are talking about a chronically activated innate immune system that is geared towards more for the trigger and less for its protective and censoring ability. You are going to have a leaky gut. You have chronic inflammation. The barrier system is going to be affected. So you're going to have leakage of materials in and outside of the gut. You also can have potential for translocation of bacteria, both normal microbiota as well as pathogens that can get systemic into the animal. The immune system or the neuroendocrine system, you're going to alter growth hormone production. That makes sense. The animal's not growing hormone because the hormones are being decreased. The enteric nervous system is going to have a negative effect on the development of neurons because, again, the whole physiology of the gut is being changed. And obviously, that last one, the oxidative status is changed. If you are inducing an inflammatory response, you are bringing in immune cells that are producing high levels of reactive oxygen nutrients, and free radicals, which not only cause damage, but change the oxidative status. So, for example, in a highly anaerobic condition like the Zika, the oxygen level goes up. All of a sudden, the anaerobes that are normally there for protective are decreased, and potential pathogens like Salmonella and Campylobacter thrive in this type of environment. They're facultative. And the same throughout the entire gut. Oxygen levels are important for which bugs can survive. Next slide, please. Okay, now let's talk about this modeling system that Gabriella developed. One of the questions we had at the beginning of her PhD in conjunction with Innovad was, can we develop a model using diet as a stimulant, as the trigger? And what should we use as a nutrient stimulant, as a nutrient trigger? And Gabriella did her master's in nutrition, and she came back, and if I the next please. She came back with, let's use rice, but use rice at a greater concentration than normal. Rice is a great energy source. It's used around the world as a feed ingredient. But Gabriella says, well, let's go above what would normally be a normal percentage of rice and use 20-30%. So we have a very high or very much excess of the NSPs, the non-starch polysaccharides. And we fed these animals for a period of 5 weeks. And every 7 days, starting at 14 days, we would euthanize some birds, open them up, and look at the intestine and do various things. Next slide, please. And what was immediately apparent is that when we opened the birds up, at the entire time that these birds were fed this rice diet, we saw no clinical or gross lesions on the intestine, inside or outside. So we took samples for histology and put them through the IC-Inside system to be able to score inflammation. Was there a histological way of finding whether there's histology there? And that's what this slide shows, is that histologically we found that with 14 to 21 days after we started feeding this, that the upper part of the small intestine, the duodenum jejunum, exhibited a inflammatory response that was very measurable, very dramatic, but was not showing clinical signs. And that the longer we fed the animals, 28, 36 days, that the lower part of the gut, the ileum, became very inflamed. And by the end, the entire gut was inflamed. So this was a very unusual that we found this temporal and spatial evolution of inflammation throughout the gut. Next slide, please. Excuse me. Now, this slide, the question of performance. What effect did this have, this chronic inflammation, subclinical inflammation, have on performance? Well, this is a preliminary experiment where we used a small number of birds and only look for the first 3 weeks at the effect. But what we did is we used two different percentages of rice, 20 and 30% here. We also had as a positive control the BMD, the bacitracin, as a antimicrobial growth promoter. And you can see that from the first 14 days, there were significant changes in both weight gain, feed intake, and feed conversion, and that it continued even more into the first 3 weeks. Now, as I said, this is preliminary data where we need to include a much larger number of birds, but it is very indicative of that subclinical, low-grade chronic inflammation does have an effect on performance. Next slide, please. Now, the question of biomarkers was a component of Gabriella's research, and just in the last 4 years, I listed some of the papers here. Now, two of them are review papers that I suggest you look at. The first, the Ducatel paper, and then the Pietro paper right in the middle, really reviewed what has been done in terms of biomarkers in poultry. The others, up to the Gabriella's, which is the last one, used various models, infection models. You see the DeMeyer paper used antibiotic growth promoters, a diet, both a bacterial cocktail as well as infection model. But what was consistent throughout a lot of these papers that people were looking at markers for gut integrity. We wanted to look at something somewhat different. We weren't necessarily looking for a leaky gut model, but looking for something that would provide us with as a marker for a long-term idea that we could predict a potential inflammatory response is going on, even if the birds did not look sick. Next slide, please. So this slide lists 4 of the markers that we ended up looking at through preliminary experiments. The first one, calprotectin, is interesting because the reason we looked at calprotectin is the definitive biomarker for inflammatory bowel disease. And not only as a marker for disease, but as a predictor for whether an individual, a human, can develop inflammatory bowel disease. So through millions and millions of samples, they were able to take serum samples from humans and predict that this person is highly susceptible or not to calprotectin. Now, one of the review papers says calprotectin is not in chickens. What's interesting is that that's true in terms of the protein. Calprotectin in humans is a 3 to 4 protein molecule. Chickens, it is single peptide, this avian MRP-126. Calprotectin is an antimicrobial peptide released by granulocytes. And we found that this was a kit that we can measure. So we looked at using an ELISA kit to measure calprotectin. Lipocalin has been used before, another antimicrobial peptide. And this one, lipocalin binds iron versus calprotectin is zinc binding. The hypoxia-inducible factor, one of the I've mentioned in the past about metabolism and taking away from the bird to feed the immune system, feed the innate system. Well, hypoxia-inducible factor as a transcription factor is a great predictor of high metabolic activity. Think about this: the immune cells are migrating into the gut, for our case, and they're migrating from a highly oxygenated environment of the blood, and they're moving into the gut, which has low oxygen level. And so they must be able to adapt to that lower oxygen level and then induce this factor, which then turns on other metabolic pathways. Specifically, HIF is a predictor and a stimulant for glycolysis, which is the main metabolic pathway of immune cells in their activation state. And the last thing we looked at was cytokines, and there's been plenty of reports in the literature about cytokines. What we did is that we found a protein array that actually measured a series of chicken cytokines production, protein production, instead of gene expression. We wanted to get somewhat away from gene expression, and simply because genes do not always show protein. And so we use this. If I can get a couple of clicks here. One more. We found that both lipocalin and HIF, we did not find significant differences in the serum, so I'm not going to talk about that, but I think this was as much an issue with the number of birds that were used, and more experiments and more birds will provide more data. So I have the next slide, please. So the first slide is looking at calprotectin in the serum of animals that were fed this high rice diet. And you can see over the period of the chronic inflammation, 14 to 28 days, that yes, indeed, the rice diet increased the amount of calprotectin that we found in the serum. Keep in mind that the control diet, remember, these are birds that are on the floor in a chicken house. Environmental stresses are there, normal stresses. It wasn't absolutely clean. That calprotectin is produced under normal situations. This is a perfectly hygienic condition, but that you induce a specific stimulus like this, we found an increase in calprotectin. Now, what I can't say yet, because we don't have enough samples, is calprotectin a predictor? Can we use levels to predict that these animals may be susceptible because of their environment to an inflammatory situation? That's going to take a lot more animals in future experiments. Next slide, please. Now, this slide talks about looking at the three different biomarkers in the excreta, in the fecal material. And we will concentrate on looking at the last two sampling periods, the 28, 36 days. Interesting, calprotectin numerically was significant, but we didn't, was numerically different, but not significant. But the earlier times, calprotectin was. So calprotectin looks like it may be a better predictor of an earlier start to a chronic inflammation. Whereas lipocalin, which we didn't find in the serum, but did find in the fecal material later, may actually be a later indicator of inflammation. And then HIF, it does prove the proof of concept that HIF was definitely significant. It is an indicator that there is a higher metabolism in the birds fed this rice diet, this trigger. But again, it's there normally in the control conditions. So, we have to establish what a baseline is, but we do find that that may be a biomarker for inflammation. Next slide, please. Now, the cytokines, what was interesting for cytokine production is that we did not see any change in what are normally considered pro-inflammatory cytokines, IL-1, IL-6, TNF. We did not find production of the cytokine. Gene expression changed, which I'm not showing, but we did not find that protein was actually produced. What we did find was these three different cytokines that one doesn't think about. And what's significant about this is that they're all involved with T lymphocytes. And if we look over to the far right at the MIP3, MIP3A was found to be significant early at 14 days. Well, MIP3A is a chemokine for T cells. And what we think, and if you look at 28, 36 days, the differences, there are, again, some numerical difference, but not statistical, in both the jejunum and the ileum. What we think is occurring is that the MIP3, if we went back and looked at, say, 7 to 10 days, we think that is the peak time that MIP3 is starting to attract T cells, that the acute inflammatory response is ending, and we are beginning this chronic inflammatory response, which is mediated by T cells, and the MIP3 is an indicator of the T cells coming in. IL-21 and IL-16 are T cell activating cytokines. So they're indicators that T cells are there and they're activated. And again, we think back to that timeline when we saw the histological changes. Remember, day 14 to 21, we found in the upper part of the gut. If you look here at 14 days, there's nothing going on. Look at IL-21, for example, and IL-16. 16 is not showing any kind of production differences in the upper part of the jejunum or the ileum early, but it then transitions at the later part that IL-16 is increased in its production. IL-21 is produced early at the time we're seeing and it's produced in the jejunum and not the ileum at the time we're seeing that inflammation in the upper part of the gut, and then it transitions down. So IL-16 and IL-21 are great indicators that T cells are coming in and mediating this chronic inflammatory response. So if I have the last slide, please. So basically just summary of the findings, this latter part of the talk. We have developed what we think is a consistent diet-based low-grade chronic inflammatory model. As I mentioned, we saw no clinical or gross lesions. Everything was histological. The preliminary data does show that this inflammation does affect performance, so we may use that as a marker as well. That we found these spatial temporal patterns, which is very, very interesting, that different parts of the gut react at different times in terms of inflammation. What does it have to do with some biomarkers, which is further experiment, and then of course these new biomarkers, and we're real excited about the idea of using metabolic markers like HIF, as well as the T cell markers. Can we show this transition from a macrophage acute inflammatory based model to the T cell model based on cytokine and on these metabolic markers like HIF and subsequent other markers that are in the pipeline? And so with that, I will end my presentation and be glad to answer any questions. Thank you very much.
Thank you, Michael, for this really dense presentation. I think we learned about how to define a healthy gut, the effects of removing AGPs, the chronic intestinal inflammation model, and also about the new biomarkers of a low-grade chronic inflammation. Valuable knowledge, I would say, in tackling chronic intestinal inflammation. I have a couple of questions here on my screen, and the first is more for clarification, and the question is, did the trial in which histological inflammation was seen include a negative control without rice in the diet?
Yes, it did. I mean, everything was compared back to a control diet. So what we showed were the differences between the rice diet and the control diet.
That's clear then. And what is the relevance of the so-called rice bran model of chronic gut inflammation compared to the real production environment?
Well, we decided that rice, because it's used as a main component of diets in at least half the world out in the Far East, that it would be a good model. It does have high levels of non-starch polysaccharides, and yet it's still highly digestible, great energy source. So it just gave us a different idea to use instead of trying to use a corn, soybean-based diet, is that we can raise the amount of rice in the diet and still be nutritious, still be balanced, and still a real world situation as we move forward and we think about corn prices and land and water usage, that rice may be a more valuable source for a chicken feed as we move forward. So it was down to that.
Yeah, that makes perfect sense. One last question before we go to the next speaker. Could environmental factors such as noxious gases such as ammonia and hydrogen sulfide, which production start in the gut, could they lead to chronic inflammation as well?
Absolutely. Yeah, absolutely. It's a situation that it's a stressor, and that stressor can lead to how cells react and can absolutely lead to certainly acute and long-term exposure to such things would definitely be potential triggers for chronic inflammation. Yes.
Well, thank you very much for explaining, Michael. For the sake of time, we want to continue with the second presentation of today. Jason Loger is Marketing Director at Innovad. As a veterinarian with many years of experience in the animal feed, additive and pharmaceutical industry, he will focus today on chronic gut inflammation, an endemic and hidden cost for your broiler production. So please share your thoughts on this important and really costly issue.
Thank you very much, Fabian. Thanks, Mike, for guiding us through exactly what chronic gut inflammation is. And thank you to everybody that has managed to join us from all over the world. As mentioned, I'm Jason Loger, Marketing Director at Innovad. And I'll talk to you a little bit about chronic gut inflammation and a potential solution that we have to solve it. If you wouldn't mind bringing up the first slide, Fabian, that would be great. Sorry, one back. Thank you. So as Mike said, chronic gut inflammation brings with it a drop in performance that's often not seen clinically, but is probably felt economically. And with Lumens, what we're trying to do is we're trying to unleash this hidden potential that's being covered by this chronic gut inflammation. Now we can go to the next slide. Thanks, Fabian. And what we need to realize is that this actually leads to a performance dilemma. We have a dilemma in our broiler production, and the reason is that we can keep increasing performance, but this will come at a cost, and some of that cost has been described by Michael Kogut. And it's these costs that we don't necessarily think about or focus on in the day-to-day running of a broiler production. Now, this dilemma plays a critical role in the development of gut inflammation and vice versa. Next slide, please. Now, if we go back a little bit into how we got to where we are, this dilemma actually started back in the 1950s, where we chose the bird on the left, or one similar to it, as the founding basis for our genetic selection. So this bird, when it was 56 days of age, 56 years would be a very old bird, but at 56 days of age, it weighed 905 grams. And then we selected this bird, and over the space of about 20 years, we managed to double it in weight to about 1,800 grams. This was in the late 70s that we reached 1,800 grams. And this was mainly due to an increase in muscle or meat. Next slide, please. By the time we got to today, another 40 odd years later, we've doubled yet again. And this has been particularly with regards to meat yield, and in particular breast meat yield, as this is the most valuable cut on the broiler. And so in the space of about 60 years, we've managed to increase the body weight by about 400%, and we have reduced FCR by about 50%. So what we've actually done is that we've increased the body weight by a compound annual growth rate of about 3.3% per year. Next slide, please. I'll just let the animation finish. So what we have on the left is an illustration of the gut or the composition of the bird. And what we see is that we have a net result where we have primarily selected birds for muscle growth. This selection for muscle growth led to a modest increase in bone mass, so skeleton to support this muscle growth, and similarly also an increase in liver weight that is also able to process all of the protein to produce this muscle. But actually, the size or efficiency of the heart and lungs hasn't changed significantly. In fact, evidence shows that the heart is relatively smaller as a proportion of the body weight than earlier generations. Similarly, the intestine has not changed significantly in size or digestive efficiency, and again, is also relatively smaller as a proportion of the body weight. Now, this imbalance has given some of the problems that we see today and continue to see in our birds. So we have ascites, we have wooden breast, we have white striping, spaghetti breast, all of which have been tied back to an insufficiency or at least an imbalance in arterial and blood supply to these tissues. And as recently as last month, an article was published in Poultry Science by Kathleen Gregory that are now showing that on the skin side, there is a new vessel that's being developed that is bridging from the skin to the breast meat, potentially as a way for the animal to try and correct this lack of supply to the breast muscle. Now, given all of these problems that we have, if we look into the heart and the lungs, it wouldn't be a big surprise to see that actually we also have problems from the supply side with regards to the intestine. And this could also be one of the reasons that we have some of these problems with gut inflammation, and probably also one of the reasons why one of the most interesting areas to focus on at this point in time in broiler production is gut health. Next slide, please, Fabian. Now, one would think that this problem would be solved by now, but actually we might be exacerbating it or making it worse in that we continue to select birds that grow faster at a younger age. And what you see on this graph is that we have the 2005 birds relative to actually each of them are relative to 1957. But what you can see is that the birds are growing faster up until day 14, maybe up until day 21, compared to the 2005 birds. And this is also illustrated by the number of days that it takes for the birds to reach 2 kg. So in 1957, it took 87 days. By 1968, it took 61 days. 2005, it was 35 days. And even in that relatively short space of time since 2005, we've reduced the number of days to reach 2 kgs by an additional 2 days. And this is a trend that is expected to continue. So not only do we have a less mature bird, we of course have a less mature gut. And this less mature gut has to absorb more feed to be able to achieve the growth performance that we're looking for. This also means that just between 2005 and 2019, within the first 14 days, we see an increase in feed intake of more than 110 grams. Next slide, please. Now, what this is doing to the bird is that this bird is in a very delicate balance. It's almost like a house of cards, if you will. And this house of cards is made up of a number of different levels of influences. So we have what I choose to call endogenic factors. So things that are going on from within the bird's immunity, gut maturity, the microbial balance, the passage rate of the feed, gut integrity, endotoxins. And by no means is this a complete list. This is just to illustrate some of the things that are going on in the bird. We then have factors We then have factors like exogenic factors that are coming from outside, things that we introduce into the bird. So feed quality, protein levels, mycotoxins, and changes in feed. And then at the top level, we have the macro environment, which is often related to management and how we manage the bird. So it's litter quality, it's ventilation, overcrowding. It could also be something like temperature, heat stress, etc. Thanks, Fabian. Now, given the number of factors, any disturbance in these factors can cause a kind of collapse of this house of cards, if you will. And when this collapses, this creates stress in the animal, and stress causes inflammation. Now, if we don't remove those stresses, what we end up with is a chronic inflammation. Now, given the number of factors that can influence and stress this animal, we believe that these birds are probably in a state of chronic inflammation through most of their lives. Now, some might think, well, this isn't really that much of a problem because actually we're getting quite good performance and no one's questioning that we are getting good performance. The question is, can it be better? Next slide, please. Now, when we have chronic inflammation, we have tissue damage. Next. And as Mike was talking about in his talk, the primary response is an innate immune response coming from granulocytes like macrophages, dendritic cells, etc. These release cytokines, and next. And these cytokines, of course, have various effects on a number of different organ systems. So if we look at muscle, we can have meat loss because of muscle catabolism. It might not necessarily be due to catabolism itself. It could also just be a redirection of resources so that you don't get an increased growth in muscle. You have stress, again, referring to the neuroendocrine axis that Mike was talking about. We have stress and stress hormones that can cause the animal to stop eating or eat less. We have all the effects that there are around leaky gut with a loss of nutrient absorption. And then finally, we also have this immune activation with energy loss. Thanks, Fabian. Now, as Mike was probably a little bit more into than I will go into here, this problem has always been there. We've just treated it differently in the past. And the way that we used to treat it was, of course, by using antibiotic growth promoters. Next. And as Mike was alluding to, we don't necessarily believe today that the bactericidal effect has been the greatest effect that we've gotten from these antibiotic growth promoters, but it has more been the anti-inflammatory effect. Next. And this is also evidenced by the microbials, the antimicrobials that we ended up using as AGPs. Because we have beta-lactams that aren't anti-inflammatory and were never used as AGP, so amoxicillin type products, similar with enrofloxacin, so from the quinolone group of or a class of antibiotics. Whereas if we look at antibiotics like tetracycline or bacitracin or virginiamycin, all of these do have anti-inflammatory effects and were used as AGPs. Next. So this also means that if you're looking to reduce antibiotics or have a production without any antibiotics ever, you need to have a component in your diet that is able to control this chronic gut inflammation. Next, please. Now, chronic gut inflammation, while no specific studies have investigated, it's probably safe to assume that most chronic inflammation probably starts in the gut. And this is probably, or this is, at least in my opinion, due to the fact that 70% of the immune system is located in the gut, and that this is an entry point for many of the triggers that lead to the four main elements of chronic gut inflammation. So the immune system is triggered by cytokines. It can be triggered by mycotoxins, enterotoxins, anti-nutritional factors like NSPs or trypsin from soya, these type of things that all promote inflammation. Once this inflammation is triggered, you get oxidative stress, you get the release of free radicals from the immune cells themselves or from the enterocytes that are being damaged. Then, of course, this leads to leaky gut because you get enterocytes that are dying, you get immune cell infiltration between enterocytes that is damaging your tight junctions. And this then gives an imbalance in the gut that gives way or an opportunity for pathogens to establish themselves, like Clostridium perfringens or other pathogens. Now, all of this added together will give you an economic loss. And this economic loss is often undetected and expresses itself as this low-grade chronic gut inflammation. Next, please. Now, when trying to study chronic gut inflammation, next. What has been done historically is kind of it's been done in these kind of settings that, if you will, are very clean settings relatively clean, low pressure, no overcrowding, etc. So if you will, almost like a 5-star chicken hotel. What you then do is you try to introduce several challenges to these animals. This can be in the form of dietary interventions, feed restriction. You can give low-level coccidia, or even introduce synthetic chemicals like dexamethasone. Now, we saw that this wasn't working, and so what we did in Innovad, next please, is that we then, instead of trying to mimic reality, we actually decided to do our trials in reality. And what we have here is a farm that we use in Belgium, 55,000 birds in a house, 48 different cages within this house. So the birds that are in the cages are all exposed to the environment that is part of a normal production. Next, please. And this is what we then call our novel model. It's under real farm conditions. We do give, if we want to challenge, we give a mild dietary intervention. We take out NSP enzymes, coccidiostats. We don't vaccinate or give any medication. Now, the interesting thing is, next please. The interesting thing is that the results are actually quite striking in the difference between what we can see. And on the left-hand side, where we have our current challenge model, so the 5-star hotel, what you can see is that in the challenged birds, they reach a dysbacteriosis score of around 3. Whereas in our trial, in the non-challenged birds, so birds that we're not giving any challenging in any way, and are generally just exposed to the environment that they're living in, they are already having a dysbacteriosis score of around 3. We then introduce a challenge, and this challenge then pushes them to a higher dysbacteriosis score. Now, what's interesting is that we can see that this is chronic because we're getting similar results. There's a slight drop, but overall you can see that you have a dysbacteriosis score that is close to the same levels whether you look at day 21 or at day 28. Next, please. Now, what we have done here is we've done 2 trials, and in these 2 trials, well, as our innovation director says, if you get a good result, in biology, don't try and do the trial again, because you're not guaranteed to get the same result again. But he's taken the chance, and he's actually done this trial twice, and he did it once in summer and once in winter. And in both cases, we saw a statistically significant improvement in body weight gain. Similarly, we saw a statistically significant improvement in feed conversion ratio. And next, and depending on the level of product that we gave to these animals, we were seeing a return of investment of around 2.5 to 4. Now, what's interesting about this trial, in particular the summer trial, is that because this was in real world circumstances, these birds were coincidentally exposed to a heat wave, because this was done in Europe in 2020, where there was a heat wave. So these birds are in a house that reached 32 degrees Celsius or 89 degrees Fahrenheit for our American friends. So even under these circumstances, Lumens was able to prove that it has a good return on investment. Next slide, please. Now, the reason that Lumens is able to do this is because it actually goes in and controls these 4 elements of chronic gut inflammation. So next, we have an anti-inflammatory effect where we reduce pro-inflammatory cytokines, and this helps to control the immune response to damages. Next, we have antioxidative effect that strengthens cellular protection and reduces this oxidative damage and thereby protects the gut cells. We have a gut barrier strengthening effect because we both protect the enterocytes, but we also have a more direct effect in the tight junction protein production. And then lastly, we have the ability to control pathogens. Now, if we add all of these components together, next, thanks, what we get is that we're able to bridge the gap, and through this multimodal action of lumens, we can help maintain gut function and profitability, performance, and of course, through that, profitability. Next. Now, the way we've measured and the way that we've seen that we have this anti-inflammatory effect is through a trial where we can show these effects by looking at the in vivo expression of messenger RNA genes that eventually will lead to the production of these cytokines. We have looked at a broad panel of these cytokines and are showing just a few here. Amongst them is TNF-alpha, a well-known tumor necrosis factor, interleukin-6, and interestingly also INOS, which is basically inducible nitric oxide synthase, which induces the expression of nitric oxide, which is known to be a pro-inflammatory mediator. And what we're seeing here is that we're seeing a significant reduction in the production of these inflammatory cytokines, and this is at a very high level, up to 210% decrease. And the researchers that are looking at this have said to us that it's unusual, or they tend to get quite excited when they see a decrease of around 30 to 50% in these type of trials. So this is a very significant effect. If we then go to the next slide, please. Here, using the same method, we looked at antioxidant production. Or actually, sorry, that's not entirely true. We looked at the production of cytoprotective proteins that help to manage the effects of oxidation. And we're looking at proteins like catalase or peroxidine that are able to reduce the oxidative species, or the protein NRF2, which helps increase cellular protection against oxidation. Again, we found some very significant effects, up to 4.5-fold increase in the expression of these genes. So we feel that we can demonstrate that lumens has a very strong cytoprotective activity in vivo. Next, please. Second to last is gut barrier function. Here again, using the same method, we looked at a number of different proteins. The first three on the graph are all transmembrane tight junction proteins, so claudine, ocludine, and then the last one is a mucin protein. And what we can see is that all of these are also increased up to 5-fold. Now thank you. This goes to explain what we see with some of our earlier trials where we did what's known as a leaky gut model, where what you give is you give this chemical called phytc-dextrin to the birds, and this phytc-dextrin, if you have a leaky gut, ends up in the bloodstream. And if you don't have a leaky gut, then of course you're able to keep this phytc-dextrin from passing into the bloodstream. So here we have quite clear evidence of what might be happening to improve gut barrier function with the help of lumens. Next, please. And then finally, we have some very interesting data that's on the way. I've put there hot off the press, but it's actually not even in press. It's very new data that we've received. And what we looked at is the total microbiome metagenomics. So basically, it's a kind of a shotgun approach where you look at everything that's going on in the gut. You're looking at somewhere between 20 and 30,000 genes. And then you're looking at which ones are upregulated and downregulated. You're looking at maybe the top 300 to try and get some kind of interpretation of what's going on. And interestingly, we can already at a preliminary level see that we have a downregulation of microbial pathogenic genes like these toxigenic E. coli, EPEC, and EHEC. We can see a downregulation of antibiotic resistance genes. For example, beta-lactam resistance genes are downregulated, tetracycline genes are downregulated. And this method also looks at various pathways where we can see a downregulation of oxidative stress pathways. So this all fits in quite nicely with the other work that we're doing and kind of shows that we have a pathogen control activity. Next, please. Now, some of you might be wondering, how is it that lumens can do all of these things? And one of the reasons that lumens works is because it's a complex synergistic formula of many different components: butyrens, medium chain fatty acids, essential oils, and plant extracts. And we know that it's synergistic because we've done an in vitro trial where what we look at is the nitric oxide production in vitro, and we're comparing it to a control, and we're looking at each of the individual components to see what their effect is. Next, please. What their effect is, where we can see that we have an effect from fatty acids, from plant extracts, each by themselves. But even when we add all of those effects together, the additive effect, there is still a gap that we can't explain, and that gap we then attribute to the synergistic effect of lumens. Next, please. So in rounding off, with lumens, we're able to bring back the performance in these birds, and it's because we have an anti-inflammatory effect, an antioxidative effect. We're able to support the gut barrier function, and we have an control pathogens. And next, please. And this means that at the end of the day, lumens brings more to the table, literally in the form of more meat on the bird at the dinner table, and also thereby figuratively by improving performance and especially your economic performance in your broiler flocks. And then for the last slide, so we believe that lumens is capable of bridging the performance gap caused by chronic gut inflammation with a proven return of investment of 2.4 to 4. And the product is available in two forms, both a liquid and a dry form. Liquid, of course, suitable for use in drinking water, should you choose to do that. So that's all that I had from my side to entertain you with for now, and I'd like to hand it over to Fabian to open the floor for questions.
Well, thank you very much, Jason. I really liked the building blocks you put in your presentation. We saw why do we have chronic gut inflammation, what can we do to alleviate the problem, and also, most importantly, what are the benefits of addressing chronic gut inflammation. So really interesting insights indeed. And I'm curious how that resonates with our audience as well. So I have the same poll question as we had before. And just see if you change your mind after the talks of Michael and Jason. The question is, what percentage of broilers do you now believe are affected by chronic gut inflammation? And you can click on the answer of your choice, which is A, less than 10%, B, less than 25%, C, up to 50%, D, over 50%, and E, all broilers or 100%. So please click on the answer of your choice and give us a couple of minutes to process the results, and then we come back to that. In the meantime, I have some questions for Jason here on my screen, also for clarification purposes. Where did you get the samples for gene expression from? Is it the liver or the muscles or?
It's the intestine. It's so basically they've taken out the entire intestine, and then from the bird. So we sacrificed the birds, and we took the entire intestine and then and sent that for sampling.
Okay, that's really clear. Another question to Jason here on my screen. What was the statistical significance of the results in the Belgium trial with increased density?
I'm assuming you're referring to the novel model, the dysbacteriosis model. And as far as I'm aware, we have a statistical significance that's less than p less than 0.05.
That's perfect. Yeah.
Good.
Okay. Last question here before we go to the results of the poll. With regards to the biomarker data, is there a single time point adequate for complete evaluation?
Well, actually, for those of you that were observant, you would see that in some of the data I was actually showing data from 35 days, and in some of the data I was actually showing data from 28 days. So we have the same data from several time points, and the trends and the effects that I showed are the same. There may be slight differences in the strength of the effect. And actually what's curious about this is that we actually took blood samples as well, and we could reflect the same trends in blood samples with regards to most of the parameters we looked at, so pro-inflammatory cytokines, etc. Well, of course, not the tight junction proteins, but the ones that you can then measure in the blood, we could see the same trends. So I guess the question maybe also for Mike here is, it's a bit of a classical chicken and the egg question here. Is it the gut that is the primary source of the inflammation that is turning systemic, or is it systemic inflammation that's affecting the gut, or is it maybe a lot more fluid than we tend to think?
So I believe it starts at the gut and works its way out. I think the idea, the fact that there's a gut liver, gut lung, gut brain axis shows that the gut is the center point, and that the inflammation flows out from there and affects the other organs.
Great, thanks, Mike. That's also kind of the feeling that we have in our organization.
Yeah. And by looking at the intestines on the one hand and doing the blood work on the other hand, you have like a double safeguard in getting the results.
Exactly. And I think that's also part of the reason why we're trying to kind of hunt, I suppose, for a biomarker, because if we can locate a biomarker or correctly identify the biomarker, then we would be able to, on a field level, identify what is the level of chronic gut inflammation in these birds. And I don't know, Mike, my feeling as well is maybe that it's maybe not just, we're not going to get a silver bullet, one biomarker that we can use, but I think we'll probably end up with a panel of different things, cytokines and proteins, etc., that will give us that indication.
Yeah, I agree. And I think where we're heading, and I think we'll get there, is that using this panel, we will be able to make a prediction of the management, what's going on a farm, whether these birds will even become, chronic inflammation become an issue or not, based on a panel of markers. So yeah, I think we're heading that way.
Right.
Well, at least interesting to move forward. About moving forward, I have the poll results in, and the question was, what percentage of broilers do you now believe are affected by chronic gut inflammation? We asked the question before both presentations and after, and there are some results. I'm not sure how significant they are, but I will have a look at my screen. At the first instance, 3% answered less than 10%, and that moved to 4% in the second question. In the first question, there was 21% less than 25%. That changed to 9%. In the up to 50% category, in the first instance, 29% answered C, and that changed to 26%, so not that much. And then over 50% came from 35 and went to 36. And the biggest change is all broilers, at which claim went from 12% to 26%. So, Jason, from your experience, are the estimates of our audience close to reality, or is the damage even greater than that?
To be very honest, I think their guess is almost as good as mine at this point in time, and this has got to do with the fact that I think that, yes, probably on some level you do have some level of chronic inflammation across most birds within a flock. It's just a question of what is the severity of those birds. And if you recall, when I showed the data around the dysbacteriosis score in the real flock, you must remember that what we're looking at there is an average of a flock. So we have an average in that flock that was at around 3. So you are probably going to have some birds that are up at around 5 or 6, and you're also going to have birds that are below, so at about 1 or 2. So the question is, for me, more, I think there could be differences between flocks, but I think that to a certain, and that the difference is in the intensity of the chronic gut inflammation. But I guess my my feeling is that it is probably in the vast majority of our birds on some level or another, just given the number of factors that can influence it.
Exactly. Your opinion on that, Mike?
Yeah, I agree. There's some inflammation that would be considered chronic. There is going to be genetic variation, susceptibility. That's why I like the idea that trying to find if we're going to come up with a panel, can we use those biomarkers to find a baseline that, okay, here's a range that gives us a predictor of normal versus birds that would be under some level of inflammation versus birds that are under high level of inflammation. And it's going to be by flock and not by individual bird basis. It's going to have to be a house by house type of thing. I don't think we're going to be able to do it anywhere near just pick 10 birds and say making total prediction. They're there. Inflammation is there. The interesting component will be identifying what that inflammation is. What's the trigger? Like you mentioned, and like we showed in the presentations, there are multiple triggers, and you're not going to be able to get rid of them all. So it's going to take something that's going to neutralize that activity, like you were talking about, that's going to be most beneficial to the animal. The other aspect I think we totally forget about is that we're talking about inflammation of a gut, and we know of at least 4 main sections of the gut, and really haven't concentrated at all on what does each one bring to the table in terms of inflammation. Certainly that progression that I talked about brought us some interesting questions. Why do we have a progression over time? Is that significant or not? Is an early measurement telling us what's going on in the upper gut versus a later measurement, later part of the life, telling us that it's reached down to the lower part of the gut? That's questions that need to be answered too.
Julie noted that there's work in progress, but when there's one thing clear for me, that now we at least have the problem on our radar, not subclinically anymore, so it's a little bit easier to tackle. So that brings us to the end of this webinar. I want to express my gratitude to Innovad, and of course to today's speakers, Michael Kogut and Jason Langer. Thank you for being here and sharing your thoughts on how to confront the issue of chronic gut inflammation. At least for my part, I've got some superb takeaways, and I'm sure you as the audience has as well. A great thank you for your questions. I really value them, and I think the speakers do too. As a last interactive action from my side, I want to point you towards the possibility to fill in a survey to help us improve future webinars. We will email you a playback link to this webinar directly after the broadcast, so you can rewatch all the information-dense presentations and, of course, share it within your network, social or otherwise. So thank you for now, and see you next time.